So these genes are nasty little parasites, and you would think the rest of the genome would turn it off. And the genome tries. They try to suppress its mode of action, but the genome doesn't always win. And these class of genes, called WTFs, are especially sneaky, especially nasty, because they're really hard to turn off. And so in this most recent paper, we looked at the ability of these genes to turn on. How are they transcriptionally controlled? How do these genes get turned on? And why can't the rest of the genome stop them from being turned on? And it turns out they're extra sneaky and have evolved a way to use a master factor in the cell. So if you stop this factor from making these genes, you'll just stop the process altogether. So there's really no way to just turn off these genes without breaking the whole system. So that's really sneaky. So that's how these WTF, selfish, nasty little parasites have been able to avoid transcriptional suppression. They can't just be easily turned off. And so our most recent paper is investigating how they're turned on and why that hasn't been broken by the rest of the genome yet. These sorts of sneaky parasites are found throughout eukaryotes, and it's known that they're going to be in conflict with the rest of the cooperative genes. The rest of the genes in the genome are going to want to stop these nasty parasites and turn them off because they're doing something bad. They're decreasing fertility. They're actively destroying the cells that don't inherit them. So it's in the best interest of the organism and the rest of the cooperative DNA to turn off these nasty parasites. So understanding how these parasites avoid being turned off, avoid what is known as suppression, is very important. And we believe studying these parasites in POMB and fission yeast will be able to help us to understand how these parasites might be acting in other organisms, including humans. So these selfish parasites, the way they work is by actively destroying cells. So if we could manipulate that killing ability to destroy cells that could be beneficial for human health, such as pests, what if we could manipulate these parasites to be expressed in mosquitoes and drive their destruction? So it's a way to control pest species by creating a gene drive system where you take a gene, like a WTF, that its job is to kill cells. If you can manipulate that and put it into mosquitoes, you could find a way to destroy certain members of that population, for example. So there's ways to take these nasty little parasites and manipulate their use for human health by creating things such as gene drives in pest species like mosquitoes. Using the WTFs themselves in a gene drive system might not happen tomorrow, but understanding how they destroy and kill cells will help us to, later on, when we want to manipulate that ability to destroy cells for our own use, such as in vectors, understanding how they work will help us to be able to utilize them potentially later in something like a gene drive system.